Okay, so good morning. So first of all, thanks to the organizers for this interesting meeting and for the kind invitation. So although this meeting is about extreme value statistics, this is not what I'll talk about today simply because I'm not working on this topic anymore. But we have worked on it like 10 years ago. So I just want to give just one slide just to tell you what we did, and in case you're interested, you can look it up. So basic idea was the following, that there is a distribution of fitness effects. So for example, that mutations come in 3 flavors: beneficial, deleterious, and neutral ones. The beneficial ones are rare. Neutral and deleterious much more common. And because they are rare, they are extreme events. So therefore, that was sort of the basic idea due to Alan Orr and other people like Joyce. So they used the extreme value theory to model the beneficial fitness effects, distribution of beneficial fitness effects. So you can do that by modeling it through this distribution, which is a generalized Pareto distribution, which is nice because it has single parameter kappa, which you can change to access all the 3 universality classes in a single, by changing single parameter. Okay, so what we did was to take this distribution of beneficial fitness effects, and we studied how that impacts the adaptation dynamics. So one of the things we saw is that if you think in terms of a model called adaptive walk, in which the population just climbs uphill, then every step, how much is the fitness difference, how much fitness increases. So that changes qualitatively depending on whether kappa is 0, positive or negative. And then we had other quantitative results as well. So if you're interested, you can look up these references. But that's something just to tell you that maybe I fit in a little bit here. Okay, but that's what I want to talk about today, because that's the sort of the latest work of ours. So I will speak about neutral genetic diversity and how that's impacted by the changing population size. And some of the work is done with Kaushik and Johari at the University of North Carolina. It is all available in bioarchive. Okay, so let's start from the beginning. What is the genetic diversity and what is this neutral genetic diversity? So let's think of the following. Suppose I take 4 of you in this room and we sequence your genome. So you are the 4 samples, and somehow I know which one is the wild type allele and which is the mutant. So wild type allele I label by 0 and the mutant by 1. Okay, and I'm interested today only on the neutral parts of the genome. So suppose we do that. So the sequencing is done, and sample number 1 has let's the first site was 0. That is wild type. Second site had a mutant, and so on. So similarly, all the 4 samples. So that's the sort of data, as far as I understand, is what people analyze. And you can do lots of things with that sort of data. Okay. So one of the simplest things that you can do is to look at pairwise diversity. So what you do is take first sequence and second sequence, and they're aligned, and then you ask the number of sites where they're different from each other. Okay. And then you do for first, third, first, fourth, and so on. So that's basically the mean having distance that you can find from this sort of data. Okay. So this is the quantity, which is a simple quantity to measure. And what do we expect? So if you assume that the mutation rates are small and the number of loci is very large, then it's a good approximation to assume that wild type only becomes mutant, because you're not going to hit the same site again. Okay. So 0 only becomes 1. And therefore, when I'm talking about the number of differences, it's simply the number of ones in this sample, right? Okay. So therefore, you're just counting the mean number of ones. And that is simply the number of individuals times the mutation probability. Okay. So that's the sort of the formula for a diploid population due to Kimura, that the mean diversity is simply 4 times n mu. So important thing here is that this diversity increases linearly with the population size. Okay, so that's fine, but this is what has been measured. So this is something which I'm showing from a recent work. This has been measured many, many times. So what has been shown here is the pairwise diversity as a function of the census population size. What you see here is for various types of organisms. We are somewhere here, I think. Okay, so what you see here is that the Kimura's result, which is linear one, it works quite well when the population size is small, okay? But for most of the data, it's way off, orders of magnitude off. So this has been a problem. This is called the Levantines paradox, who measured these things almost 50 years ago. And this has been a big question in the subject. why is the neutral genetic diversity so low? And it sort of, makes sense because you see here, you have low population where genetic drift or stochasticity is high. That's the dominant force. So that works because that's what the theory is assuming. But over here, there are other forces as well. There's not just genetic drift, selection, migration, and other stuff. So therefore, that has not been taken care of, right, over here. So no wonder it doesn't work. But one still wants to understand that we are thinking of neutral things. So how is that working out? Why is this diversity so low? Okay, so this many mechanisms have been proposed. Question, yes, sorry. Yeah. So So this per people, there's something called biodiversity map. So using that, this author managed to find the range of these organisms. So therefore, you know the area in which they hang around. And then there is a phenomenological law called Damout's law, which relates population density to the mass of the organism. So therefore, using that, you know population density, you can measure the mass, you know the population density, you know the range, and therefore you can find the number. But it's sort of approximate, but there's no doubt that there is qualitative difference. I mean, that's not doubtful at all. Any other questions so far? Okay, good. Okay, so these mechanisms people have thought about because 50-year-old problem. There are lots of mechanisms to explain this, but till date we don't have a quantitative resolution of this problem. So all these mechanisms that have been proposed, they all reduce diversity, but not by this orders of magnitude. Okay, so this is the sort of question, but we will keep this Leontief's paradox in the back of our mind and just think about these mechanisms on their own because they're important and interesting. Okay. So I will just introduce one of these mechanisms which I want to describe in detail. So this is called selective sweep. So let's just take an extreme case to understand it. So what we're thinking about is the following. So suppose we have just a two locus problem, just two sites. And these two sites are linked physically. They just hang around together always. This is asexual. Okay. So there are two loci. First locus is selected one. It's over here, the first one, which has little a or capital A. And the second locus is neutral. So initially, I have neutral diversity because I have both types of alleles at the neutral locus. So at some point, if a beneficial mutation occurs at the second locus, and it occurred with the little b allele, okay, and provided this capital A does not get lost, because of fluctuation it can get lost, provided it doesn't get lost, it will spread through the population, right? Okay, but you see what happened. Because this guy is beneficial, it spread, but then because of the linkage, it dragged along that little b along with it. So I started with this population with both capital B and little b, but now I have only little b's left. So in this extreme case, neutral genetic diversity is 0. Right? So that's the sort of the basic idea here, that the linked selection will reduce genetic diversity. Okay, so that's the key idea. It sweeps away the genetic diversity. Okay, but this was assuming there was these were always linked, but recombination occurs, which breaks the linkage. So there's a lot of work which has been done when linkage can be broken. So, however, much less is known, relatively less is known when population is asexual, and that's what I'm interested in. Okay, so basically we generalize the basic idea that I told you. So, but we have now not just two loci, but infinite number of loci. And what we have here is this initially population, which is neutral here, and the selected site, beneficial mutation occurred, and then it spread through the population, and then because there are many, many loci, which are neutral ones, little a and capital A, so they will come and go because of mutation, they will keep coming, the new neutral loci, the mutants and the neutral loci, and they will keep going away because of absorptions. Okay, so then you'll get some non-trivial genetic diversity. But what I want to point out is that if you look in this part, in this subpopulation, you can ignore the selected site and model its effect as though you have just the neutral population whose size is growing. Okay, so the size of this population, neutral population, is growing, something that we've been hearing all this week about growing populations. So that's what sort of little bit connection to Luria-Dilbruck. Okay, so I have a neutral subpopulation which is growing. However, it reaches a finite size n. So it's not just expanding away. There's a limit, there's a carrying capacity in our model. Okay? And we are interested in asking these various measures of nutrigenetic diversity. The quantity that I want to focus on is slightly complicated than what I described in the beginning. It's called mean sojourn time, or related quantity called site frequency spectrum. So it's a little bit complicated, but let me explain using an example. Again, we look at the sort of data I showed you earlier. So suppose we go site by site. Look at the first site. At some point, maybe, so I'm looking at the sample today. At some point, maybe a 1 appeared somewhere, which is my mutant allele, but it got lost because of fluctuations. At site number 6, 1 did not get lost, it got fixed in the population. First site and 6 sites are uninteresting because there's no diversity there. But at other sites, you see there's 3 sites at which we have 1 mutant, there are 2 sites which we have 2 mutants, and 0 sites which we have 3 mutants. So that kind of histogram, that's called a SFS, or site frequency spectrum. And that, from this kind of data, is obviously can be measured. So this is the quantity which can be measured. People do it routinely, and that's why we are interested in this. And other measures of diversity, like when I introduced in the beginning, they can be related to this. So that's the quantity which I want to focus on, and I will give formulas, etc., about this object. So if it's new to you, do look up at the recording and see if you still have questions. I'm happy to answer later. Okay, so what is known about this? So because it's a sort of classical quantity, what is known about it? So one classical answer is that if you look at large times where mutations are coming and leaving because of absorption, then there's a sort of a mutation drift pattern which is reached. If I have a neutral population of fixed size, then sojourn time, or the SFS, the site frequency spectrum, it decreases as 1 by x, x being the mutant frequency. So that's a well-known result. But if you have an exponentially growing population without a limit, it just grows, then SFS decreases much faster. It grows as 1 by x square. Okay, these are the known results due to Kimura and Duret. What we have is also an exponentially growing population, but it's sort of, at least in the simplest case, it increases logistically. It increases and then saturates. The result due to Duret is obtained using a branching process, which, as you know, assumes the resident population to have infinite size. I have a finite size population, so I have to do a little bit more work. I cannot use branching process, but I'll use a Fokker-Planck equation. Okay, so let me say here right now that we have to say a little bit more about how I'm modeling this population size growth. I'm going to right now assume it's growing deterministically because I'm assuming right now strong selection. So they're slightly complicated looking equations, but you are physicists, so I think it's fine. Okay, so it can be shown that this mean sojourn time, it obeys this equation. This is second derivative term, and we have this x into 1 minus inside the second derivative. And then we have these coefficients, which are also time dependent, where is time dependence n grows logistically. And so that's the equation. But as you know, PDEs, we have to supplement with initial condition, boundary conditions. So I'll not give much detail, but I should just mention that this is to be supplemented with a boundary injection, because you have to keep putting mutants, otherwise the absorption will occur. So that's a sort of the PDE that we solve. And it's solvable thanks to Kimura's previous work, who showed that this operator, the eigenvalue equation for this operator, is a hypergeometric equation. Okay, this is a Gauss hypergeometric equation. So therefore, it can be solved quite easily. So the sojourn time is this wave function, which is a hypergeometric function. And then there's a time-dependent part, which looks a little bit complicated. Remember, I Schrodinger equation, right? But it's a function of this object. There's an integral simply because n of t can be absorbed over here in this derivative, right? So that's what this term is. Okay, so in principle, we have an exact solution for the SFS for population which is growing in any manner. It doesn't need to be exponentially large. So we have, in general, we have the answer. But this solution is very, at least to me, it's very opaque. It's not clear what's happening because there's an integral and blah, blah. Okay, so what we have been able to do is to develop some approximations for it so that at least the solution is writable in terms of relatively simpler or more elementary functions. Okay, so which is shown here. The solution over there is approximately given by this expression. Okay, and here you have a green line, which is my expression, and red are the simulation results for certain parameters, and it works quite well. So that's the formula. But what is it teaching us? So this shows that the SFS, this quantity of interest, it has a crossover from Kimura's result, which decays as 1 by x at low frequencies, to 1 by x square, which is Duret's result at large frequencies. And this crossover occurs at 1 by ns. So let's just try to understand this result a bit. So if you have no selection, then we have in the Kimura's regime. That means crossover frequency has gone to infinity, and I expect 1 by x all through. On the other hand, if I have a very large population, expect Duret's result all over, right, over there. So therefore, there's a crossover in between. So that's not unexpected. I mean, that will happen. More physically, at low frequencies, population is expanding, but mutant frequency is very low. It is not aware that, your population is expanding. It will not see these boundaries. So it will behave as though it's a constant size population. And therefore, you have this 1 by x. As, mutant frequency increases, it will be more sensitive to increasing population size, and therefore you have a crossover to 1 by x square. Okay? So let me also mention that the asymptotic answers are sort of, you can argue and one can understand it, but I'm not giving you only the asymptotics. I'm giving you the full function here. The full answer is what we are giving you, and not just that, I have not explained that, but I'm also giving you sojourn time as a function of time. Okay, so this is a full time dependent quantity. To make our problem even more biologically relevant, we also considered inbreeding and dominance as selected sites because it's more relevant for biological applications. And we find that these laws of 1 by x and 1 by x square are universal in the sense that these exponents continue to hold even with these other things that we add. Okay. Yeah. Hypergeometric, etc., are approximated, and then the sum is also approximately dealt with. Yeah, yeah. The simulations, in fact. Yeah, yeah, yeah. Yeah. So it works in certain limits. So certain parameters, if they are very low, etc., it doesn't work that well. Yeah. So we know. I mean, I can tell you what approximations have gone in. Yeah. We know that, of course. Yeah. 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 There are functions of parameters and time and so on, and I didn't want to write it, but it's in the paper. Yes, that's a 4n mu or 2n mu. Yeah. But the W and V, they also have other stuff like N as well. N, S, I, H, dominance, etc. So they are there, but still it's a relatively simpler result. Any other questions? Yes. Yeah. So this I'm showing you at the end of the sweep. But yes, XC also depends upon time. That's correct. Yeah, I assumed it's deterministic. It's expanding population, but it's increasing logistically. It's deterministically growing population and saturates. That time scale is determined by selection coefficient, and I'm assuming NS is large. So it increases to NS, N, and over time scale is 1 by S. So I'm increasing, NS is large. That's what you assume. Any question? Because I want to shift gears a bit. Okay, fine. So this is all okay. But what I'm really excited about, and I want to share this, is the following. In this discussion, I assume strong selection. But of course, neutral sweeps also occur. Selection can also be weak, which means the population size, which is increasing, which is changing, is not going to change deterministically. It will change stochastically. It will fluctuate. Okay. So that's the question that has not been studied much in the literature. And if anybody knows, please do let me know. I would be very interested. Okay. So let's consider this problem in general. So what do we need for that? So therefore, if I just write the answer schematically here. Okay, so we have a wave function and then we have this object. Then we have to also average over stochastic population trajectory, which I'm denoting by these symbols, right? Okay, so we need to do this average, but it's all the time till t. So this is, of course, difficult because it's over all the paths till time t. But there are many ways of thinking about it. But one way which I like is that we can express this in terms of the cumulants of the stochastically changing population size. So we have the mean of the inverse population size, 2-point function, 3-point. I mean, there's a whole infinite hierarchy here. And this is kind of useful for the following reason, which I'll explain. But because effective population size is mentioned, I will just take 1 minute to explain that. So what is this? I mean, so we have this complicated function and so on. But in this context here, population size changing. So many times population biologists try to think like this. If you have a complex problem, replace it by a simpler problem, but which has all parameters sort of constant. So it will be changing space, time, etc. But think of it as though it's a constant size problem or constant some effective parameter problem. So let me demonstrate that, how that works. So in this case, if I look at this object, so this is the exact expansion. If somehow I drop all the correlations, okay, so all these terms are gone, and further I assume stationary process, so therefore this integral is easy and it comes out over here. So this object is equal to what you would expect in a constant size population. So you equate this equal to this, and this n effective is nothing but the harmonic mean. Okay, so this has been replaced. If I can neglect correlations, it has been replaced by a problem whose effective population size is the harmonic mean with respect to this stochastically changing population sizes distribution. Okay, and this again you will see a lot in literature. It's done a lot, but of course you cannot neglect correlations in general. It will work sometimes, but not always. So I prefer not to just think of effective population size, just do the problem completely. Okay, so correlations are important, and we have again heard of that in other talks, in other contexts. So correlations we have to take into account, otherwise it's sort of this very simple answer which is not going to work. Okay, so we need to consider such an expansion, and there are correlations, etc. And how do we deal with that? So again, if I go back to sweeps, which is what motivated to look at stochastically changing population size, for neutral sweeps, it doesn't seem possible to do these things exactly. I have some answers, approximation one, but it's sort of approximate. So, but it might be nice just to play with some problem in which you can do everything exactly. So I'll just give you a toy model. It's not necessarily relevant to evolution or something, but it's exactly doable. So let me quickly introduce a model to you. It's a very simple model. So we have the usual Wright-Fisher process. So, but the population size is changing between two values, n1 and n2. And n1 and n2 flip between each other at a certain rate, gamma. So that is called a telegraph process. It's a switch, on and off. If you think in discrete, if you're somebody who knows Markov chains, etc., then it's just a two-state Markov chain. But now I'm considering in the continuous space and time. Okay, so we are considering a case where the population size and when n2 are changing at a rate gamma, and because gamma is the only gamma inverse is the only time scale, the population size will remain correlated over this time scale gamma inverse. Right? Okay. So you have this Wright-Fisher process in which the progeny are chosen, offspring are chosen from the previous generation by equal likelihood. For example, these two are chosen to be, this is a mutant, it's chosen with probability 1/3, and at some point, the population size became 5. So therefore, you do the sampling 5 times, but you choose this again with the probability 1/3, the dark blue one. So that's how you can do the simulation. So we introduce the mutant in the stationary state of the telegraph process and run the simulation. But this model can be solved exactly, and let me just very briefly explain what the Fokker-Planck equations are. So it matters, you have to write these equations for the probability that the mutant is starting whether in n1 or n2. So that's what this subscript is. So let me just say it again. So pi is the probability that the mutant starting in frequency p in population size ni reaches x in time t. Okay. So I'm writing a backward Fokker-Planck equations. So these are the terms which you will generally write for your favorite Moran process or Wright-Fisher process. And these are the terms that are coming due to telegraph process. What we have here are the coupled Fokker-Planck equations with appropriate boundary conditions and initial conditions are exactly solvable. And you can relate it to the sojourn time. Okay, so you can look up the solution if you like, but let's see what do we again learn from this. So we have just two time scales in this problem, population size and the switching rate. So imagine the switching is very, very slow. So the population size remains constant, and then 2 years later it will switch. So if the mutant came in N1, it will basically see that constant population size, right? So that's the size, the constant size it will see. So that's a slow switching case, in which case you will see the sojourn time to be the usual 1 by x decay, and the prefactor turns out to be 2. But on the other hand, gamma is large, so the population the size is switching very, very rapidly. But if it's switching rapidly, that means correlations have decayed very fast. And I've already told you, when correlations decay fast, then in that case, harmonic mean comes into play. So then if you work out, then the sojourn time is again decaying as 1 by x, but the prefactor is changed, and this harmonic mean comes into play. So you have these two limiting cases when you have separation of time scales. These are easy answers because it's just 1 by x and only the prefactors change. That's easy. But what is interesting is, and the real question to my mind is, what happens in between when the two time scales are overlapping? So that's where I think the value of these kind of problems is, because you can give exact answers where these time scales are not separated. So this exact solution shows the approximation simply because something like sum is replaced by integral. That's sort of approximation. Otherwise, it's just exact. So the solution turns out to be this for the SFS and for the sojourn time, and it's shown over here. Let me just explain what it's saying. It's saying that at low frequencies, this SFS, mean sojourn time behaves as though it's in the slow switching regime. Okay, and then at high frequency, it behaves as though it's in the population, the rapid switching regime. We are in the moderate switching regime, but at low frequency and high frequency, it is behaving like slow or rapid. And that makes sense, because at low frequencies, the mutant has not spent sufficient time in the population. So it's just going to see only one population size. It's not aware that it's going to switch, right? Okay, so therefore it sees as though switching is slow. But at high frequencies, it has seen several changes, and therefore it behaves as though it's in the rapid switching regime. Okay? So you have a function at low frequencies over here, and over here, and then in between, at intermediate regimes, there is this other, some other behavior, which is all contained in this result. Okay, so I think that's basically I wanted to say. Oh yeah, sorry, I forgot. So the crossover occurs again here between these two regimes at a frequency which is this function of this dimensionless variable n gamma inverse. Okay, so that's all I think I wanted to say here, which is that I gave you just two examples where population size was changing, once deterministically and another time stochastically, and mean sojourn time, which is a quantity of interest because people measure it. It is not proportional to 1 by x in general. And we expect, however, we expect that there will be crossovers on overlapping time scales. And that's where I think my interest lies. So I'm interested in understanding if population size fluctuates or selection coefficient fluctuates, or in general, both fluctuate. Then what can we say about the diversity? How is it affected when time scales overlap? So that's what I'm interested in. Thank you very much.